We're here at the Rothamsted Research Library, which houses the Long Ashton annual reports which document the initiation of the National Willow Collection. The National Willow Collection was created in 1923 following the impact of the First World War on basket making production. Basket willow was identified by the War Office as a strategic resource. The committee recommended appointing a willows officer to support the industry in conducting scientific research into basket willows. Important uses of willows in the war effort included shell panniers, baskets for carrier pigeons, observational balloon baskets, and chairs and spinal carriages for the wounded soldiers, as well as medical panniers. From 1923, basket willow varieties were collected and secured in a living collection at Long Ashton, just north of the willow growing area on the Somerset levels, where it continued to be augmented with interesting germplasm. The multifunctional end uses, which mirror the versatile genus Salix, are represented in the National Willow Collection. Applications include amenity planting, bank stabilisation, cricket bats, windbreaks, paper pulp, and providing wood chip for bioenergy feedstocks. This unique germplasm resource has been used to underpin a number of research programmes here at Rothamsted Research. Cuttings from the National Willow Collection are made available to members of the public and basket makers each year. The National Willow Collection is a fantastic resource that underpins lots of research here. So this is one of the largest collections of Salix in the world, hosted here at Rothamsted. And we've got 1,500 different accessions of willow. So the morphology within the genus Salix is very diverse, and here we've got two really extreme examples. This has got really large leaves. Its common name is the magnolia leaf willow, or Salix magnifica, and it's got much broader leaves in this ovate shape, which are really in contrast to these tightly curled leaves of Salix babylonica, variety annularis. You can see they're very, very different, and they sum up some of the amazing diversity that we have within the genus Salix. This is Salix daphnoides, native to Europe and Russia, and it's got these lovely violet stems. It has been coppiced here, but in nature it would be a small tree. This is Salix melanostachys, which is a much smaller growing shrub and produces lovely black catkins in the winter. In the coming weeks, the yellow anthers will have emerged from these black catkins, which is a real spectacle. This is Salix tortuosa, or the corkscrew willow, which is an ornamental willow, famed for its curly stems used by flower arrangers, and it can look really nice as winter stems as well and provide colour in the garden. So this is Salix alba, a tree species. We're seeing it in its coppice form at the moment, but if left to be a tree, it would be over 30 metres tall when mature. And this one's actually the cricket bat willow. It's a female willow, which is grown for cricket bats and also clogs. This is Salix purpurea usney, and it has really fine short stems that would make a really good choice for some really fine basket making. So commercial basketry's concentrated on the Somerset levels now, and has done since the First World War, but there were other areas of the country that grew lots of willow, and Lancashire was a big willow growing area. So this is Salix purpurea, or the purple willow, and this makes a really good fine basket willow because it's got very slender, pliable stems. This one's called Lancashire Dix, and it was grown extensively before the war in the Maudsley region of Lancashire. This is Salix viminalis, or the osier, and it's got these classic lanceolate-shaped leaves, or spear-shaped leaves, and traditionally it's been used for hurdle production, but more recently it's been used to make living sculptures or large sculptures. This is a Salix purpurea, Salix viminalis hybrid, and you can see the intermediate traits between the two species. So it's good for bigger basket work or hurdle work. Okay, so this is Salix triandra, or the almond leaf willow, and this specific variety is called Black Mall, and this one variety makes up around 80% of the commercial basket area planted on the Somerset levels. It's really prized by basket makers as it's very pliable and it's got hardly any pith and it's very hard wearing.
I'm Ian Shield, an agronomist and plant breeder, and for the last 15 or so years I've been involved in the bioenergy project at Rothamsted, looking at how to grow energy crops but also in breeding willow for the energy crop market. So willow is of interest to us because it's amongst the fastest growing trees that we have in the UK, and this is particularly so when you grow it as short rotation coppice, so grow it on a short harvesting cycle. We produce a wood fuel, primarily wood chip, and this can replace coal, oil or gas in a combustion type heat or heat and power type energy market. So the benefits of growing willow as an energy crop, first of all, as I said, replacing coal, oil and gas, and with much better greenhouse gas profile from a renewable rather than a fossil fuel. Willow can be grown on land that has few other options, which is quite a positive. There are areas of the country where landowners don't have a lot of options as to what they do, and this is a new one. There's some good biodiversity effects, and particularly when you mix willow into the environment with other farming activities, boosting the overall environmental status of the landscape. The inputs into willow are not terribly large, particularly when compared to arable crops that we might conventionally grow in many areas, which makes them relatively low cost and low input to grow. Willow is grown by propagating from stems, so we plant short sections of stem from usually a one-year-old plant, which have been cut and made into what we call cuttings. They're planted in rows so that they can be mechanically harvested, and there's about 15,000 cuttings planted per hectare. The crop is sensitive to weed competition in the early phase, certainly in the first year, but at the end of the first year it's cut, usually not harvested or used, but it's cut to get that classic multi-shoot form of a coppiced tree, and then it's left to grow for usually about 3 years and harvested on a 3-year cycle. Some fertilizer is used to boost the crop and to ensure that we get a commercially viable yield.
Willows are a dioecious species, so individual plants are either male or female. Willows are brought into the glass houses so controlled crosses can take place. Once seeds are produced, the generated progeny from the crosses is subject to a number of rounds of selection and screening to assess its suitability for its end uses. For SRC biomass willow, it can take over 10 years from making the initial cross to releasing a new variety, having progressed the material through 4 rounds of selection. The same breeding techniques can be used to select other traits of interest for other applications, such as the high-value compounds.
My name's Steve Hanley. I'm a molecular geneticist at Rothamsted Research, and I have responsibility for the molecular aspects of the willow breeding programme. We're using genetics to increase the efficiency of the willow breeding process by improving our understanding of the genetic control of the traits we aim to improve. Examples being biomass yield, disease resistance, plant architecture, the production of specific compounds and so on. By doing this, we can help the breeders to predict how the offspring of a particular cross might perform in the field, and this allows us to make more informed choices about which crosses to actually make. We do this by looking for associations between what a willow looks like in the field and variations in its DNA, its genome, And for this we've generated populations of several thousand trees that we've intensively studied and measured in field experiments over several years. So by combining the performance data from these trials with state-of-the-art DNA sequencing analysis, we have developed a catalogue of associations between particular differences in the DNA and variation in our target traits. In conventional plant breeding, a breeder makes a cross and selects offspring that perform better than the parents. However, in reality, this is often only a small proportion of the plants produced. So by screening the DNA of new lines early in the laboratory, we can quickly select for promising offsprings at the seedling stage, which stops us from having to raise and assess all the plants in the field. And this can be both expensive and time-consuming. This approach also allows us to make a better choice of parental combination, again potentially saving time and resource. The huge amount of diversity in willow, both at the trait and DNA level, is helping us improve our understanding of the fundamental biology of plants, how they work, how they interact with environments, and so on. This research is also allowing the ongoing discovery of novel genes that affect a wide range of traits, both of biological and economic significance.
My name's Jane Ward. I'm a chemist, a natural product chemist, and we're looking at using willow as a source of novel compounds for potential products for the bioeconomy. And the National Willow Collection really underpins this work, and that's due to its chemical diversity that's present in the plant. Many of the compounds that are used in resins or adhesives or the production of those materials are actually derived from fossil fuels, and this is a dwindling resource. So if we can get the types of chemicals that are used for this starting of these processes from a tree like willow, it actually provides a much more sustainable source for the future. So the National Willow Collection completely underpins this area of science.
I'm Mike Beale and I'm leading the chemistry research into willow at Rothamsted Research. So the discovery of aspirin came from one or two willow species. We now in the National Willow Collection have a huge gold mine of chemistry that we can exploit for the development of new medicines. And we are effectively revisiting the pharmacology of willow using modern high throughput techniques such as NMR spectroscopy and mass spectrometry. We extract the various willow types and we analyse them with our equipment, and we identify new molecules, and we work the structures out of those new molecules, but also at the same time we are testing them against cancer cell lines or in assays to do with neurological diseases. So then we relate bioactivity to individual molecules within the willow. Since the discovery of aspirin, the potential of willow as a medicine has been relatively unexplored. We now have the opportunity with the National Willow Collection and its hugely diverse chemistry to actually revisit the pharmacology of willow. And this, we believe, will give us a huge resource for the development of new medicines in the future, particularly for diseases like cancer and neurological diseases such as Alzheimer's disease.